much of the genetic code we share with other beings. Whether it's a monkey or a banana, whether it's a bird or a cat. But more importantly is how similar we are to each other. And yet, we have our own individual characteristics and traits which make us unique and completely different from one another. This is the phenomenon of epigenetics, modification in the expression of the genetic code without altering the structure. Good evening, everybody. It's a great pleasure and honor to be here. Unlike the typical helical strand of DNA that was the Watson and Crick model, the epigenome consists of a series of small markers or tags which get attached to different points on the surface of the DNA. They do not change the structure, but alter which part of the genetic code is expressed and which part has to be suppressed. Simply put, it brings about changes in the phenotype, but not in the genotype. This explains why identical twins with an identical genetic code grow and age differently. You might have one twin, being a marathon runner at 55, and his brother having a serious cardiac problem at the same time. There are many processes involved in epigenesis, but what is relevant and important to us are the processes of DNA methylation and histone modification. This is a really long topic and a very short time, so I'm trying to make it as simple as possible. In the embryo, epigenetic programming happens essentially at two stages. One, in the primordial germ cell, and then in the embryo just after fertilization, that is the pre-implantation embryo. This is of critical importance because most of the procedures in an ART cycle are performed when the dynamic state of programming and reprogramming is happening in the gametes and the embryos. Consequently, any disruption anywhere in the cycle, whether it's with hormone stimulation, oxygen levels, culture media, redox potentials, ex vivo manipulation, can lead to altered gene expression, gene suppression, and imprint disorders. So to say, an individual's epigenome is believed to be the basis of its health and the origin of its diseased state. It has been postulated that most of the procedures in an ART cycle can interfere with epigenetic programming, ovulation induction, IVM, micromanipulation, embryo culture, you name it. Ovulation induction not only overrides the natural process of oocyte selection, growth, and maturation, but also may possibly recruit poor quality oocytes which were originally meant for destruction. Accelerated growth following gonadotrophin administration can affect the process of DNA methylation. This may result in the release of M2 oocytes with an incomplete or labile genomic imprint. This is now a documented cause of imprint disorders which have been associated with ART, mainly the Beckwith-Wiedemann syndrome. In fact, Beckwith-Wiedemann syndrome is said to occur in about 1 in 3,000 of all children born from IVF and ICSI. Some form of, it need not be a florid syndrome, but some form of the syndrome. Embryo culture is believed to produce the same effects. Disrupted methylation in the zygote leads to growth arrest and early embryo loss. Innumerable studies have been carried out on the effect of culture media and birth weight. Some say yes, some say no. There is no consensus. And I believe there are a few studies which consistently say that vitro life is the only media said to increase birth weight by 200 to 300 grams. The jury is still out on this one. The confusion in the culture media scenario has led Eshrey to set out recommendations, I think in last year or the year before that. They've been asking for transparency in the composition of the culture media from commercial manufacturers, requesting labs to ensure quality management systems are in place to make sure the media are stored and utilized properly. Clinical follow-up of offspring, and maintenance of a national registry for culture media, identify and document which culture media is used. While we know now that epigenetics and the genome are responsible for, well, the embryo's health and eventually our health, more research is still essential. Findings are useless unless data is collected. Data is useless without analysis. Analysis is useless without dissemination of information and formulation of guidelines. As of now, there is no consensus yet on how to counsel patients regarding the epigenetic consequences of ART. But what do we know? We know factors that can possibly affect the epigenome: diet, exercise, stress, medication, chemicals, herbicides, pesticides, environmental pollutants. We also know that epigenetic changes are transgenerational. We know that the changes can be carried down 2 to 3 generations down the line. Life experiences of our parents as children, whether good or bad, and the choices they made as adults, whether good or bad, have already affected our individual epigenomes. In fact, a couple of years ago, there was a very controversial study which documented the incidence of depression in grandchildren of Holocaust survivors. Last year, Andre Fisher, on a more positive note, actually demonstrated modified learning ability in offspring by subjecting the dads to environmental enrichment, diet, exercise, cognitive conditioning. So a simple way, send dads to boot camp. You'll get smarter kids. What can we do to make ART safe? We are increasing the number of cycles, we are increasing the success rates, but we have to make it safe, not only for the patients now, but for the generations ahead. Standardized manufacture of culture media, standardized follow-up methodology, minimal stimulation protocols, and a holistic approach. What we know today is just the tip of the iceberg. As we learn more about how the epigenome influences us and how ART influences the epigenome, a number of exciting possibilities, both therapeutic and diagnostic, are opening up. In fact, currently, I think last year, there was a startup in the US which got funded, which tests for the epigenome of sperms, the methylation of sperms, which is going to tell us whether the sperms are capable of fertilization and whether they're able to produce a healthy, normal individual. The test is whoppingly expensive, $500. But they claim that it doesn't have any, the samples are sent from home via mail order, and they're analyzed and you get the reports online. And they claim that they don't need any specific storage methods for the sperm, and they're ready to do a semen testing for the Neanderthal man also. I end over here. Thank you very much for a patient hearing.